I'm going to hand it over to Nathaniel Virgo to introduce our keynote.
Okay, so in the early days of ALIFE, Chris Langton described artificial life as the study of life as it could be, in contrast to biology as the study of life as it is. But a big part of that, maybe in the overlap between those two questions, is whether there are universal laws that govern biology. So that's what Chris Kempes is going to be talking about. He's a professor at the Santa Fe Institute and also external faculty at the Complexity Science Hub in Vienna. And he does work that is highly informed by biology, but that is directly relevant to artificial life as an enterprise, and it's really fascinating work. I'm really looking forward to it. So yeah, take it away, Chris.
Yeah, thank you so much, Nathaniel. I also really want to thank the organizers and the Society for the invitation to be here. I also want to thank the organizers for what's been a fantastic conference so far. Maybe we can all give them a quick round of applause. It's something they don't get a lot, and yeah. So as Nathaniel said, I'm interested in the universal laws of life. And so today I want to focus on one tiny little question, which is just, are there universal laws of life? It's a small question, it's an old question. It's something I think should be a central question for all of us and for our entire field moving forward. So just to situate this a little bit, what do I mean by a law of life? Well, the sort of thing I'm really talking about is the type of thing that we have in physics. So here are sort of different types of triumphs in the history of physics. What's amazing about physics is that we are able to observe the world on our own planet, extract basic principles and laws, and then use those to interpret the rest of the universe, right? And what that really means is that there's a set of laws we can count on anywhere in the universe, and we apply those over and over again. So in this case, here are sort of two triumphs. We can, although in a very complicated way, model the entire dynamics of the sun, everything that's happening with heliophysics. That's all based on different principles that we extracted from observing our own world and solar system and the cosmos. And then this amazing triumph of measuring a gravitational wave produced by two colliding black holes through this interferometer at LIGO is another case where the types of theories we built on Earth apply everywhere in the universe, even to things incredibly far away. So this is sort of a triumph of a certain sort of law-like perspective. And the same is true in chemistry. Now, while chemistry is much more complicated, it's exceptionally combinatorial, and there's lots of details we still haven't figured out. For some of the basic rules, we know how to apply them again at the universal scale. So we can look at distant nebulae and interpret what chemicals are in the gas clouds that we see there, right? So again, local rules that we can apply anywhere in the universe. And the question really is, do we have these sorts of things for life? We need to, because as we start to look for life in the universe, or build new types of life in the lab, or debate about whether synthetic life or in silico life is actually life, we need some sense of laws that help guide us in that search and guide those questions. But typically for biology, what we think about is something like this. This is a really beautiful and amazing photo of diatoms, roughly at one size scale, so all these cells are about the same size. So this is both narrow in size space and narrow in taxonomic space, and yet here we see this amazing diversity. We see amazing diversity in the morphology of these different organisms. We also see amazing diversity in the physiology and biochemistry if we went inside these different cells. And so inspired by this picture, what we typically say about life is that the only way to understand a species, a trait, or some physiological function is through the detailed, highly contingent, highly historical evolutionary history of an organism, right? And I just want to mention that this is true. This perspective is definitely true for certain questions in biology. This is the only way to answer certain things about some organisms. And there will definitely be non-universal pieces of biology, and in fact, we should be interested by that and embrace that wonderful and interesting diversity. But this is also true for some types of questions, observations, scales, and problems. And what I want to highlight today is that there are other types of questions that are more law-like, where this isn't the only answer to understanding life or biology. So let me introduce a schema first that will situate all the things I talk about today. This is something that David Krakauer and I worked on a few years ago. We called this the multiple paths to multiple life. And in this framework, what we described is sort of 3 different layers of life. At this base layer, you have what we call the material layer. And this is really the type of perspective we've typically had about the history of organisms. So you have some origin of life in a particular material. That origin of life then undergoes some complicated evolutionary process with all sorts of branching, and we emerge this sort of phylogeny. And typically what we're interested in biology is reconstructing that history and asking exactly how the origin of life progressed to the set of species we have today. But something that's interesting is that we don't often talk about the fact that these materials could radically differ across different origins of life. In fact, this is a central debate in questions of origin of life in astrobiology, is just how different could all of the materials be. But we should allow that across the universe, the materials could vary quite a lot, and we could have radically different biochemistries than the one we see on our own planet. We also talked about how in this space, you could have evolution in one material, like this green material, that suddenly has a different evolution evolved out of that in a different material. So for example, AI, ALIFE, in silico life suddenly is evolving in silicon. And that is a different material than the carbon-based chemistry all of us use. And so this is sort of an interesting case where you can have one origin of life suddenly invent a new material that then has a different origin of life. And this is really what our multiple paths to multiple life are. In this paper, we argue that things like culture and language really should be viewed as separate origins of life in new materials, things like human brains. And that's not such a radical proposal for this group, but it is for some communities, so we wanted to note that. Okay, so here we have this highly diverse, highly contingent, highly historical perspective of organisms. And then we argue that there's a layer up from this that we call the space of constraints. So here what we're doing is we're using the laws of physics, the laws of chemistry, to sort of collapse what might be true for these materials into more condensed spaces of life. So think about the laws of diffusion or the laws of gravity. Organisms everywhere have to deal with those constraints. And so that might mean that, for example, 2 radically different materials get projected into the same constraint space, say around diffusion, on different planets across the universe. Now, there still could be strongly different environmental conditions on different planets, such that the space constraints or the constraint spaces don't overlap. And so we still allow for some diversity in this level, and perhaps even some of the engineered constraints that we have for artificial life or ALIFE don't necessarily overlap with these physical constraints, because it's different sorts of problems that you have in that world. But this still has some diversity, and again, we're after the universal, and so maybe there's a higher level that we can project all of these into. We call this the space of optimization principles. The idea here is that things like evolution, which is really a learning process, an optimization process through a lot of randomness over a long amount of time, things like the error threshold, ideas that are very familiar to this community, there may be a set of such principles that actually collapse all of the life we see across the universe into one very narrow space. Now, perhaps artificial life doesn't overlap with that space. It's a debate we need to talk about and think about. We think it does. We think there are certain principles that would project this into this space, but for completeness, we sort of left it outside just in case. Okay, so I want to talk for a bit about this middle level. It's the place where I think we've done the most concrete work, is thinking about what happens for the constraints. These are things we know easily from physics and chemistry. And so we should just see how far we can go with this perspective, how much we can say about life just from focusing on constraints. And then at the end, I'll come back up to this more general level. In this constraint space, these are the sorts of things I'm talking about. So this is really a remarkable plot produced by ecologists about 2 decades ago. And what this plots is the logarithmic mass of an organism against its biomass production rate. So this is how many grams of biomass it produces per individual per unit time. And amazingly, if we zoom out to the largest perspective of life, where we collapse all of life's diversity into just size against this parameter, we see roughly one relationship. That's sort of remarkable, right? So to a physicist, this looks like a law-like thing. And to a biologist, that says that if I zoom out on evolution, there seems to be a small set of constraints that matter to all organisms and are organizing their behavior across all these orders of magnitude. It's always important to note in these plots that there's tremendous variation around this curve, right? So this could be 2 orders of magnitude or more, in the scatter from this organism to that organism, and that could be due to environmental features, or it could be due to historical contingencies in the evolution of these different species. So this plot allows both for the huge richness and diversity that we see for organisms, but then also average behavior at the largest scale of evolution or life. And what we know about these relationships is that typically you can derive them from focusing on a small set of constraints. So typically you can write down just the dominant physical constraints for organisms of a particular category, co-optimize those, and predict these non-trivial scaling relationships. Here, for example, the exponent is 3/4. That's certainly quite non-trivial and takes a little bit of work to predict. And so here's our framework for that, which is to say if you have some trait of interest and you're measuring the performance of that trait and you're looking at that against body size, you could have these two different physical constraints, both of which matter to an organism, but these constraints matter differently at different scales. And so when you optimize that trait under these constraints, at this scale, only constraint one matters. This scale, only constraint 2 matters. And in between, you're co-optimizing them, and you could get sort of arbitrarily complex relationships, telling you how an organism's performance should change with body size. Amazingly, we don't often see many curves like this. Typically, we see power laws, and there's sort of good reasons for that. But for completeness, this is sort of the picture we have in our mind. And this is those physical boundaries of evolution that I had in my title slide, which is just to say that organisms are doing all of these random walks, they're evolving randomly, and they keep bumping into these constraints that they have to co-optimize on, and that gives us sort of average behavior as we go across many orders of magnitude and body size. Okay, this has been a bit abstract. So let's summarize what we think from here, those being that we'll see scaling when the same physical constraints dominate and affect diverse organisms across scale, and traits are sufficiently independent of each other. So this is just to say all the time you have a trait you'd like to optimize and you can't because it depends on some other trait, and that trait depends on different constraints, and then you won't get nice scaling relationships. Sometimes this isn't true, and it certainly isn't true for macrophysiology, so often these are the background assumptions we have. But to make this really concrete, let's take a case study of the smallest organisms. So let's zoom in on bacteria and just ask, how is this sort of general framework or principle playing out for these organisms? And here there are really sort of two mysteries I'd like to focus on that inspired a lot of work for us. And the first mystery is that if I go across these major evolutionary transitions where life is getting more complicated, I see shifts in these scaling relationships. Now I'm plotting metabolic rates, so the total watts used by an organism against its size and volume space rather than mass space. And we see that as you go from the prokaryotes to the unicellular eukaryotes to the small multicellular organisms, we see distinct shifts in these scaling relationships. We go from something that's superlinear in bacteria to something that's roughly linear scaling in the unicellular eukaryotes to something that is very sublinear, this 3/4 power scaling in multicellulars, and that goes all the way out to us and whales. But what's happening for bacteria? Why this very steep increase in metabolic rate? So that's mystery one. And mystery two is that if we look at bacteria and we sort of dig into the physiology, a few years ago we'd spotted that if you plot the total protein abundance or protein concentration in a cell, that abundance is going down slower than the total volume of an organism, meaning the concentrations are becoming more and more dilute. So this is just a different way of plotting that, where this is enzyme concentration against cell volume. It's really the same data, just converting units. And so this seems sort of striking, right? As cells get bigger, they have less protein per unit volume, they seem to be getting dilute. And so just to put that in pictorial terms, it means that small cells are sort of these densely packed encapsulations of proteins sort of nearly touching each other. And then as they get big, all of these things dilute out and you get this big sort of water-filled sac for larger and larger cells. So what's going on with these two different mysteries? And the thing to remember is sort of a classic thing that's been realized in the last 50 years, which is that life for bacteria is quite different in terms of the physics, right? So here's a very famous paper by Purcell called Life at Low Reynolds Numbers. And what he really points out in this paper is that if you zoom in on bacteria, their physics is a totally different world than the one we live in, right? So it's the equivalent, as he put it, of us swimming in molasses, right, or honey, right? So the physics are totally different. They're in this low Reynolds world where they just don't get much inertia. Everything is dominated by viscosity. And so we can expand that idea a little bit and think about what's happening inside a cell. And here, there's something we need to recognize about the strange world of bacteria, and it's all about diffusivity. So at our scale, we could double the concentration in this room, and diffusivity doesn't change at all. And so you can do that for many orders of magnitude in concentration, where you keep increasing concentration and diffusivity doesn't change. But then there's this packed regime where every increase in diffusivity comes with a decrease, sorry, every increase in concentration comes with a decrease in diffusivity according to a power law. And so now as you move out in concentration space, you're making things less and less diffusive, right? So this is the world that bacteria live in. E. coli are actually sort of here, and smaller cells are down here, as we'll show. And so in this regime, what we can do is write down the reaction diffusion dynamics for a cell with a simple addition that isn't often included, which is that the enzyme concentration Z now affects this diffusivity. And what that means is that we now have a well-defined optimum, right? So as I increase enzyme concentration, if I go too high, I decrease the diffusivity, the cell becomes diffusion limited, and that's a real problem for the cell. And if I go too low on concentration, then I just don't have enough enzymes to drive the kinetics of the system, right? So I don't have enough enzymes to actually do the reactions. And so what that means is there's this optimum where I haven't yet fully killed my diffusion, and I still have enough enzymes to run the kinetics of the cell. When we write down what this optimum is, it depends on a few physical parameters and the size of the system. And that amazingly now gives us a prediction for how this enzyme concentration would scale with cell volume. So here's the optimal enzyme concentration according to the model I just laid out. These are all, D naught is just the basal diffusion scaling from that power law of diffusion against concentration. This is the equilibrium constant. K is the summary of the kinetics. And so you see that this relationship depends on a few physical parameters and then the radius of the cell to the 4/5, which is just volume of the cell to the -4/15. Right, so this is this really highly non-trivial exponent. It's what comes out of the theory. We'd always said this data is about -1/3. Amazingly, it's actually -4/15, and that agrees pretty well with data. So the best fit is this dashed line, and then this is prediction from the theory given all the physical constants and the exponent that we predict. So this tells us why cells are getting more dilute. We can do lots of other things like predict what the sort of average enzyme property should be. This is looking at K across all enzymes, all metabolic enzymes in lots and lots of different bacteria. And this is what we predict the median of that distribution should be. And that agrees really well. Sorry, the dashed line is what we predict, and this is the actual median. And so we do a good job of also capturing what this average property of the enzyme should be. Okay, so that's great. That's mystery one. Why do cells become more dilute? They do so to manage kinetics against diffusion. And now we can also understand metabolic rate by just integrating, taking this Z optimum, integrating the total power production of cells, and now we predict that the metabolic scaling of cells should be roughly 11/15. That's not this superlinear thing that I talked about. You have to add to this an extra constraint, which is that at the small end, the cell needs significant volume for DNA and other macromolecules. We'll come back to that. But if you put all of those together, you do recover this superlinear scaling of bacteria. So we think we've solved sort of both mysteries there and interpreted what's happening here. And the thing I really want to highlight is that these are just basic properties of enzyme kinetics and diffusion. And so these are things we really expect to hold anywhere. And so our strong proposal here is that if you went to another planet and you looked at how the metabolic rate of cells, even cells with radically different biochemistry, scaled with cell volume, we think it should be this curve. The intercept could change a little bit based on different features of the enzymes. Maybe the enzymes aren't as efficient. But we think this exponent should be true for any planet where you measure life. This is something I'm willing to bet on sort of across the universe because of the constraints that go into this. That could be, now you could ask, well, when would that break? It could break early in evolution if evolution hasn't had enough time to achieve some of these optimizations on the physical constraints. But given sufficient time, we really think this is the relationship that simple cells are headed towards. Okay, great. So I want to talk a little bit about how we can use some of these relationships and sort of build up to other features of organisms and life history. And here, what we did is to say, if we know the total metabolism of an organism, we can partition that into growth plus maintenance. And since we know the scaling of this, maybe we can start to say something general about growth of cells of different size. Now, the way to write down this relationship is just you have some metabolic scaling equaling biosynthesis cost. So this is how much energy it takes to create a unit of mass times the rate of mass production of an organism. And then we have a maintenance cost, which is how much energy it takes to maintain an existing unit of biomass times how much biomass you actually have. You can rearrange this for dm/dt. You have differences of scaling relationships in this. And so you get sort of a fairly complicated growth trajectory out of this, but something that's still closed form. And this really just depends on a few simple bioenergetic parameters. And amazingly, this does a good job of predicting how the growth rate of populations changes across body mass or cell mass for both the bacteria and the unicellular eukaryotes. And these subtle differences in the scaling exponents I was talking about before tell you that as bacteria get bigger, they should grow faster and faster. And as unicellular eukaryotes grow bigger and bigger, they should get slower and slower in their specific growth rate. That's nice, but the most interesting thing about this plot are these two asymptotes that you see. Because that relationship I showed is a nonlinear budget, there's a scale at which the maintenance metabolism, just the repair costs, become the entire metabolism of an organism. And when that happens, you can't grow. And that happens for the smallest bacteria and the largest unicellular eukaryotes, right? And we'll talk about the smallest bacteria in a second, but you can see for the unicellular eukaryotes that this asymptote occurs right where you stop seeing large unicellular eukaryotes and you start seeing the small multicellular eukaryotes. So we think this predicts why there's a major evolutionary transition at this scale. It's that this is becoming a real constraint. It's becoming a wall or a boundary to evolution. And you need to do some transformation in architecture to get over that. And we could talk, find me at the break, we could talk endlessly about all the things that happen here around multicellularity that get you over this wall. But the point is we know where the wall comes from. And at the small end, it's that same boundary. It's the same place where maintenance becomes everything a cell is doing. And this predicts the smallest possible bacteria. Amazingly, this does a good job of anticipating what were found as the new world record holder smallest bacteria a couple years later. This is really beautiful work out of Jill Banfield's lab where they found these groundwater microbes that are now the smallest known bacteria. And you can see they go from about a third the size we're predicting up to basically exactly the scale that we're predicting. That sort of variation is expected. If we plotted the error bars around this, there's some fuzz in what the smallest boundary should be, and that fuzz agrees with this prediction, right? So we think now there's a reason why life or cells can't get much smaller than a certain scale. And indeed, this is where we see the transition to viruses. You get viruses smaller than this, but not cells with their own metabolism. Great, so this also left us with a mystery, which is what's happening for the large bacteria. As they get bigger, they grow faster and faster. They seem to have more metabolic power. Why do we get a transition to unicellular eukaryotes at all? Life seems good for the large bacteria. And we, in this original paper, said, well, it must be something about some basic biochemical rate that just can't keep up. Sure, you could drive this metabolic power faster, but maybe the ribosomes or the DNA replication or any number of other rates just can't keep up with that. And so we decided to dig into that. And when you do that, you can say, we know the total protein concentration in a cell. And from that, coupled with this growth rate, we can write down a model for the total production economy of a cell, where you can say this is the rate of change in the number of ribosomes over a cell cycle. It's basically a base rate of processing a base pair times how many ribosomes you have, divided by the length of all the base pairs you need to make all the ribosomal proteins. Gamma is just how many of the transcripts in the cell are dedicated to making ribosomal proteins opposed to all the rest of the proteins. And then you have a specific decay term. You can write down a similar budget for the number of proteins. And now it's just the length of the protein could shift. And it's all the ribosomes not making ribosomal proteins make the rest of the proteins in the cell. And when we put these two together, we get a bound on how many ribosomes you need as a function of the number of proteins and this growth rate scaling, and then just a bunch of constants that don't matter so much. We know both of these scalings from the two things I mentioned before, and when we put all that together, we can then predict how many ribosomes you need as a function of cell size. So you can see at the small end, there's almost no ribosomes in the cell. The small cells have about 100 ribosomes. It's sort of a vanishingly small amount. And then as cells get bigger, they grow faster and faster, demanding more ribosomes. And at some point, you would need more ribosomes than can fit in the cell. We call this the ribosome catastrophe. It's basically the cell growth rate starts to run up against the ribosome replication rate. And that means that you need more and more ribosomes per unit volume, and that just simply can't happen at some point. And so you have this sort of issue. So now we think we understand what's bounding the largest cells at something like this. And we can also expand out to talk about all the things that change in bacteria across the range of cell sizes. So in the smallest bacteria, the cell is mostly DNA expressed proteins inside the cellular envelope. And then as cells get bigger, they become more and more RNA content. And at the largest end, they're sort of dominated by RNA. So this is interesting because it says not all bacteria are the same. Not all bacteria have the same resource requirements. This has big implications for ecology and evolution and all sorts of things that we'll get into in a second. I want to step back for a second and say that I've been making a sort of strong pitch for all the things I've talked about so far being universal. And for maybe the diffusion and kinetics story, you might believe me on that. But for this story, maybe you don't. Because you could say, well, I know all sorts of detailed things that might be contingent about sort of all of these different elements of the cell. So prove to me that any of this is universal. And I'd say some of it is guaranteed not to be, but perhaps the ribosome might be, and here's a good reason why. So as many people will know in this room, Landauer proposed that for any computational process, you can write down the simple entropy transformations and use that to bound how much energy a computational process takes. And we can view the ribosome as a simple computation, one that takes an unordered set of letters and writes those into one very specific string. This has a really simple entropy transformation. And what this does is bound how much energy this process takes in general. It's really bounding string writing for any system, independent of whether it's the ribosome or not. And what we find is that you would need about 10 to the minus 20 joules per letter, joules per amino acid in this case, to run this process. We know how much energy the ribosome actually uses, and that looks like this. So it's about 10 to the minus 19 joules per amino acid. So the ribosome is 25 times less efficient than the ultimate physical limit. So it's 25 times less efficient than any device performing string writing. You might say, well, 25 seems pretty bad, but just for reference, all of our computers, everyone who's taking notes, is writing strings at 100 million times worse than this bound, right? So even after 80 years of Moore's law, we're still not anywhere close to pushing up against what the ribosome does. Furthermore, if you back out on the ribosome and say, well, you're only talking about sort of one type of entropy here, you're talking about the informational entropy of this process, but the cell also has to find all of these amino acids in 3 space and then sort of bind them into one of these strings. And if you consider that part of the process, you get that the ribosome is maybe only 4 times worse than this bound, or right up against the bound in some sort of indistinguishable way, right? So it looks like the ribosome is pretty close to being about the best device we could have for string writing. And that's really interesting. It says that even if you started to ask what's contingent about the story I've told, maybe you can't make the ribosome much better. We tried to summarize different things like this in a recent review paper that I want to point you towards. This is a paper with Ricard Soleil and a bunch of people who all happen to be at SFI at the same time. And what we talked about is really what are some of the constraints that might be fundamental to living systems across the universe. And we summarized things like thermodynamic engines, this linear information carrier I just talked about. We talk about this bound on the cost of information, which is what I just went through, and so forth and so on. And one of the things we talked about is maybe cells are minimal units. Maybe you do need cells in some sense. There's big debates about that. But here too is a place where we can start to dig in on what's essential for cells or what the bounds on cells might be. And we can start with the ribosome. So for example, we can play this game where you say, well, what if life discovered a much better, again, I think that's hard based on what I was just saying, or much worse ribosome? Well, you can see that if life got a much better ribosome, say one that was just 100 times faster, you would get many orders of magnitude increase in the possibilities for cell size. That's probably unlikely given these Landauer bounds we were just talking about. But life certainly could find a much worse ribosome. There's no guarantee that you initially find effective or efficient catalysis. And if the ribosome were, say, 100 times slower, it's actually 40 times slower, this is the 100 times line, you couldn't encapsulate life at all, right? So if you keep all of the other elements of biochemistry exactly the same, and you just make the ribosome 40 times slower, you can't get that perhaps free-living metabolism into an encapsulation. To make that really explicit, this is plotting the maximum cell volume that you could have against this normalized ribosome rate, which is just how many base pairs it can process per second for its size, which you're measuring in length units. You could also do that in volume units, right? And you can see that as you sweep across this parameter regime, you get radically bigger and bigger cells, and that's important. You could also put this in growth rate units and say, for any enzyme I think is essential to some autocatalysis, I could look at catalytic rate against the volume fraction that I use in the cell for that bit of the autocatalytic cycle I'm interested in. And now I get a whole family of growth rates as a function of those two parameters. And some of these you might say are way too fast or way too slow. This red line is the reference for E. coli. And now if I add a loss rate to an environment, which every environment comes through with either from sinking or decay or any number of other losses, predation, now I have the maximum volume fraction rate that I can have as a function of catalytic rate. And this will come with a growth rate associated with it. So given a death rate D, I can have catalytic rates where I can't actually get any of the molecule into the cell that I need, right? Because I would just need too many of them. And so this is a nice space that starts to rule out different proto-metabolisms that you could find and say there's just no way to encapsulate them if they're too slow because I would need too many of them. I also want to step back a little bit and ask sort of what applications do these scaling relationships have, right? A bit ago I was saying we can use these for ecology and evolution and perhaps astrobiology. I just want to make that explicit. So here's a very famous relationship that will be familiar to many people, the Redfield ratio. And what this is that Redfield went across the global oceans, looked at organic matter, and looked at the phosphorus against the nitrogen, and he found that you have this linear relationship telling you there's a constant ratio of nitrogen to phosphorus sort of for all life, even though how much you have can change by many orders of magnitude. It's interesting. Many people in the astrobiology community have said this is a target for life detection, right? Maybe since this is so regular across our own planet, maybe we can go anywhere and just look for nitrogen to phosphorus. But that comes with a challenge, because we don't know if different evolutions of life would use the same molecules that we have in our own biochemistry. Even if they're similar molecules, those molecules could differ in the slight amounts of nitrogen and phosphorus. So there's no guarantee that this ratio is constant. So we wanted to sort of explore that. So to do that, the first thing we need to do is figure out where this relationship comes from. How do you build up to such a relationship? And we said, well, in our model, we know how the total abundances of all of the molecules in the cell change with size. We know how to count up the elements in each of those molecules. And so now we can just do a simple accounting for cells of different size and get a total elemental abundance at each cell volume. And what that gives us is this highly non-trivial, non-monotonic relationship between nitrogen to phosphorus as a function of cell volume. And this curve is the Redfield ratio through the center. Oh, by the way, the two curves are just differences between gram-negative and gram-positive bacteria, which from the non-biologist is just differences in the membranes. And what you see is that almost no bacterium is exactly on Redfield, right? Most bacteria are sort of far away from it. E. coli-sized things roughly are sort of close to Redfield, but many exceed and many are below. And so how do we get this global ecosystem level Redfield ratio? And the answer is that you have to think about abundances. So for a long time, we've known that there are also these abundance laws. If you plot cell volume against the total number of cells, in a size bin, we get these power law relationships. And so what we can do is just do a simple accounting where we integrate the elements per cell against how many cells you have per environment. And when you basically convolve these two relationships, you get that red field pops out of that. Right? So red field is this really non-trivial combination of how many cells you have and then how nitrogen to phosphorus changes with cell size. Sort of interesting. But now that we have this, we can start to play with it. We can start to introduce deviations to this overall curve. One thing that's also worth noting is that you can put these relationships into a simple chemostat model. And here then you could look at environmental nitrogen and phosphorus against cellular nitrogen to phosphorus. And you see that as cells get bigger, they tend to be, as cells get bigger, which are these curves, they tend to be farther away from the environment at any scale. And so this is sort of interesting. Another way to put that is to make this plot where you have a cell to environment ratio just plotted against. So I shouldn't explain this. This is just a normalization of this curve. But the point is really that as you get bigger cells, you move away from the environmental concentrations. And so our point was this in itself starts to become a sort of biosignature because you could go to any fluid environment. You could just measure particles of different size and ask, do you see stoichiometric shifts? That's that non-monotonic N to P relationship. And then the other thing you could do is start to ask, do you see systematic shifts in the ratio of the particle to the fluid abundance for any fluid sample as you go to larger and larger particle sizes? But again, this was based on sort of two relationships that you might imagine shifting. One is this abundance curve with cell size. The other is this N to P relationship with cell size. We said, well, all of this could shift under a different evolution of life. Let's play that game in a simple model. And what we find is that you can make this huge space of lots of different reasonable parameter variations and ask how often does the cell level resource abundance look like the environmental resource abundance as a ratio. And this green line is just where the environment and the particles look identical in terms of their stoichiometry. The orange point is what we see on Earth. And so what you see is for lots of different parameter sweeps, and I won't go into the sort of details of this, it's a large space. It's just a very narrow manifold in that space where you have environments and cells looking identical. Typically, you would be able to tell the particle apart from the environment. So this is another sort of very general indication of life. You go to any environment and you just ask, are the particles floating in a fluid characteristically different in their stoichiometry from the fluid itself? And even if you don't know what the biochemistry is, that technique should work. This is sort of a blind way to look for life in a fluid sample. This has really nice applications to Enceladus. Recently, Cassini flew through the plumes coming out of the pole of Enceladus and used the cosmic dust analyzer to get a sense of what the nitrogen and phosphorus abundances look like in that plume, which tells us what they should look like inside the liquid ocean that's below this frozen ice sheet. And here the answer is sort of interesting. Ammonium looks pretty well constrained in terms of its molar abundances, but there's some uncertainty in how much the phosphate could vary. And if we put these two together, we're getting an N to P ratio of something that's below red field to something that's roughly 1 million, so much, much higher than red field. This seems pretty unconstrained. Is there anything we can say about what this might mean for Enceladus? So what we can do in our general framework is just build up distributions under arbitrary evolutions of life to ask where do these numbers sit. And so here's a posterior probability of the elemental N to P. This is the elemental N to P in different planets, sweeping over all of our parameter space, and then this is just the distribution of those. And then here's that range of Enceladus values that I was mentioning before. And here's the range of values we see on Earth with the dashed line being red field, right? And what's exciting is that Enceladus, if we just calculate the overlap in this space, captures about 30% of the probability. And if we allow Earth to do the same thing, just asking, well, we could get variation in the measurements we had from Earth from a single sample measurement, which is possible, then we see that in this distribution, there's sort of a 30% chance that these measurements are consistent with a living world. And if we did the same thing for Earth, just using one measurement in a blind way, we would get about the same probability, right? So in this framework, Enceladus doesn't necessarily look less living than Earth. I want to point to some other really exciting work that's been done recently. This is a team that looked at the energetics for a methanogen living on Enceladus, did the same sort of probability estimation. Amazingly, they get about 32% probability for life on Enceladus. And so this is all sort of pointing to a world that we should investigate more, I think. A place that is at least not ruled out. You know, these numbers could be 10 to the minus 20 in terms of probability. I think this points to a richness and a sort of excitement that we should look at in more and more detail. So everything I've talked about so far has sort of lived at this middle constraint space. And for the last little bit of this talk, I want to pop up and talk about this sort of higher level of optimization principle, sort of more abstract constraints for life. And here we can start with cells again and ask questions about, can we abstract any of the ideas that we had about biochemistry? Work that we did, this is work I did with Sarah Walker's group, was to look at genomes across the diversity of all life and ask questions about their broad enzyme functionality. So there's a really nice classification scheme to turn every enzyme into one of these broad categories of function where you can see these enzymes do different things. Some of them change the handedness of molecules by moving something around inside a molecule. Others attach to molecules, others cleave molecules. These are all sort of very general, broad chemical reactions, independent of the details. And here too, we can look at how these things scale with system size. And we do that across all of life. We find that each of these categories has roughly the same scaling relationship across all of the different domains that we looked at, all the way up to the metagenome level, which is really an ecosystem level property. Summarizing those exponents looks like this, and you can see for certain categories of these enzymes, the exponents aren't really changing across any of these different categorizations or summaries or coarse grainings of life. And this is very different than the physical story I told. The physics we know are shifting at each of those major evolutionary transitions. That doesn't seem to be happening for enzymes. Enzymes seem to sort of have a single scaling relationship across all the categories. At present, this is entirely phenomenological. We have no idea in terms of details where these exponents come from or why they have the values they have. But our hypothesis is that what's really happening is that this looks more like different types of computational and abstract operations, right? So if you think about putting two molecules together, that can be thought of as a concatenation of a string, for example. You can think about cleaving off parts of it, rearranging strings. And for some reason, this more sort of operational perspective on life seems to be more universal. This is a big target for theory. We're trying to understand where these exponents come from, but at least gives us hope that there's something even less contingent than what I've already described. Because the other things at least depend on physical constraints. These seem not to depend on physical constraints, but instead on some set of abstract constraints that we haven't identified yet. So that's one place where we rise up to this higher level. Another place is, and a question that we've been really excited about recently, which is just how far can we take the concept of an organism? And one example of that is to think about human organizations as organisms, right? So we've been thinking a lot about companies as organisms. And for lots of different categorizations of a company, there's no reason not to say it's an organism, right? It's a unified entity that has goals, trying to survive in a competitive world, competing with other companies in an ecosystem context, changing in time, right? This sounds like many of the things we want for an organism. Companies don't really replicate and pass on a genome in that sort of sense, but they do pass on information through people who leave abandoned or bankrupt companies for other companies, people who move between companies and carry knowledge. And so we think this is actually a pretty good model for organisms. And so we've been excited about trying to apply some of our organism growth perspectives to companies as a sort of more abstract, more general form of life. Companies also have these really nice scaling relationships. So now if we look at total assets as the size of a company is sort of the measure of the body, and we start looking at different features like the sales, liabilities, net incomes, all of these different financial features scale with the size of the company. And we can interpret this in terms of a resource flow with an organism, just like we did for energy being partitioned between growth and maintenance. And here we just get a slightly different budget. And that budget is basically the rate of change in assets, and every accountant on the planet would tell you this, is just income plus the rate of change in the liabilities. Pretty simple. But each of these terms, because of these scaling relationships, has a dependence on cell size. And so we can rewrite this equation as different scaling terms of the number of assets. And then when this liability exponent is close to 1, which it is for US companies, we get this very simple growth law that looks roughly like assets going like time to the 6th power. This is kind of interesting. We weren't expecting this time to the 6th. It's a very strange exponent. And you could say, well, that's really fast growth. It is really fast growth, but it's still slower than an exponential. All power laws are slower than an exponential. And what that means is that if we plot this prediction for growth against the whole market, we see that young companies grow faster than the market, and then old companies start to be overtaken by this exponential growth of the market. Right? So most of the advantages for young companies, and then big companies start to have a harder and harder time, and many of the deaths are sort of happening here. Interestingly, this also allows us to evaluate individual companies. So you can see that this is Coca-Cola. So if anyone buys a Coca-Cola from the vending machine, which is just right out the door, you're helping this curve. And what this really shows is that Coca-Cola is sort of one of the most average companies in the entire market. And Warren Buffett says this, right? Warren Buffett says you should just buy Coca-Cola and hold on to it for 40 years. He's sort of right. You're likely to get sort of average returns of the whole market from doing so. This is Apple, which has this crazy fast growth. And this is a plastic company that never realizes a profit, is sort of born and then just continually decays until it dies, which is interesting. So this gives us a perspective on trying to understand the performance of individual companies. But on average, as you can see, if we average across the market, all of these different sort of fluctuations are leading to this average growth curve that we have at the overall scale. Now you could say, you know, if I'm really trying to take this organism concept further, and I'm looking at this sort of new category of life, and I'm calling that companies, shouldn't we get some new insight about this different type of organism? And our model actually shows what that new insight is, and it's just that companies have debt. And this really is the key to adjusting those growth dynamics. We spent a long time as a team talking about what other things might have debt in biology, and we couldn't think of a good example. I mean, for a while we were saying, well, maybe, you know, if there's young that are being protected by adults and fed by adults, maybe that's sort of a genome lending to roughly the same genome to be passed, you know, on in the next generation, but not really, right? It's not really a good concept of debt where I can borrow from the ecosystem and then pay back the ecosystem in time for things that aren't very closely related, right? And this seems to be something really unique to companies as an organism. By the way, if anyone has a great debt example in biology, please come talk to me. It's something we've really tried to find and don't think there's a good analogy for. And that means that when we see differences in debt, because again, this roughly linear scaling of liabilities in the US is driving exactly this time to the 6, it means that when we see differences in debt like there is in the Chinese market, which has superlinear increase in liabilities with company size, that should come with different types of growth relationships. And indeed, when you have more debt in the system, what you get instead is this accelerating growth curve where you can see the largest Chinese companies are really accelerating in growth. And so this choice at a system level to give more debt to larger companies comes with a different sort of growth dynamic. It should be noted, by the way, that if you have debt that's much less than linear, you can also get curves that saturate and reach adult sizes just like all of us do. And that would be a case that would look more like a biological organism. It's not true for all the different markets that we've looked at. I just want to acknowledge for this company work, which is exceptionally new, that this really came from a team of really diverse perspectives. So these three people and myself, or these two people and myself, are physicists. But Veronica is an economist at the business school in Barcelona. Rui is a complex systems scientist, and Marcus is an archaeologist, anthropologist, and ecologist, right? And so it was really a lot of sort of interesting ideas and debates from different people within this team that led to this framework. So just to conclude, I want to bring us back to this framework and say that I think there's really a lot of interesting things we can say about this constraints level. We're beginning to say more and more about this optimization level. And the proposal I would make for this community is that we really need more conversations between these two levels, right? So much of ALIFE, and I really agree with this perspective, is about trying to understand what principles of life could be like in the absence of certain constraints. How different could life be if we don't have the constraints that we have on Earth? And that's really important. But I'll say that a lot of the interesting things that happens for biology is defined by the constraints. So I think at very least we should be conscious that every time we build a novel simulation for an organism in a new context, we're trying to get new types of features for life, we should just be aware of all the types of constraints we've put into that. Is it finite resources? Is it a finite population? Is it spatial structure? All of these things are constraints. And I think it's really the interplay between these two that help us understand the types of life that we're going to get. I'll conclude. I have a huge list of collaborators on all these papers. Most of their names have been on slides, but for the sort of high conceptual level that I've been tracing through this talk, I really want to acknowledge these 4 people: David Krakauer, Mimi Cole, Ricard Soleil, and Jeffrey West, who I've thought through a lot of these sort of high-level concepts with as thought partners. Set of really amazing funding sources, including some specific collaborations with inside NASA, which has been really wonderful. And with that, I'd be happy to take some questions and discussions. I can't get away from the idea that all I'm doing in my work is just making really bad Kandinskys. So this is a really good Kandinsky, and this is a really bad Kandinsky. So thank you for your time.
Cool. We're going to take about 5 minutes for questions. I guess I'm gonna try and I'll alternate between Slido and the room. I think there was somebody. Okay, this one. Yeah, I've got to give you the mic, don't
I? Thank you again for the talk. It's super fun to see something attack this problem from so many scales and so many angles. I wonder though, when I look at you know, especially so when you do a lot of these comparisons in like a log plot or a log plot, you know, things. I wonder how many times something will just start to like kind of shrink onto the norm, and you're sort of losing the variance by doing that sort of compression onto, you know, a log or a log, where if you evaluate that variance one step above, you have a lot more, maybe lose the significance of convergence that you're finding at that certain level of compression.
Yeah, it's a great question. We always like to say, you know, logs hide all sins, which they do, right? So there's a couple things to say about that. One is that often we do have really strong statistical support for what the exponent should be. And there, what we're really trying to do with the exponent is test the prediction of a certain set of mechanisms. So I see that more as a feedback to say, did we get the 0th order most dominant mechanisms, right? But to your point, there's a lot to learn from the higher order moments of those distributions, a lot to learn about the deviations of individual organisms. In other parts of this theoretical space, we've done a lot of that. So for example, you could say, well, temperature. If I add temperature, how much do I collapse the variation? Quite a lot. You can ask questions about drought resistance in plants. If I add sort of a consideration of arid versus non-arid environments, how much do I collapse the scatter? Again, we can do that in a theoretical way. Just adding that to our optimization, you collapse the scatter even more. And so I think it is a tool to just sort of march through a ordering of mechanisms. For this sort of work, I also think the debate is about the mechanisms. And I didn't present it here, but for each of these scaling relationships, sometimes there's quite a lot of debate about what the dominant mechanisms to derive the relationship are. I think that's a really productive debate. It's one that happened in physics for a long time. And there, what you have to do is really ask questions about which is the most parsimonious set of mechanisms that produces the most number of predictions across scaling relationships. And there's certain theories that give you a whole host of organism features all at once, and then you can test all of those scaling relationships in concert. That really is starting to look like a compact theory to me, and so I'm excited about that. But it's a perfect question, something we often don't talk about so much. Thanks.
Hey, Chris. Yeah, really interesting talk. I guess I'm curious how much these models concord with, I guess, more recent synthetic cellular and ribosome systems. Whether it's like minimal mycoplasma from Craig Venter, or tethered or engineered ribosomes from Mike Jewett, or even some of these cell-free protein synthesis systems. I'm curious, how much those end up concording with each other.
Yeah. So interestingly, the synthetic cells agree with what we have in terms of expected growth rate, size of the genome, all those sorts of things at those small scales. There's probably good physical reasons from what we're saying. In the cell-free systems, what's interesting there is that you've radically changed the volume, right? And so you're now doing things in a batch culture. That's going to have different effects, and I think it's something we need to think about. And I've been working with a bunch of experimental synthetic biologists to try and do some of that, to sort of trace where that story is going and why. But it's really important to think about. Hopefully, my hope for these sorts of mechanisms is that they become engineering tools, right? That's what we want from these sort of principles, is that like physics, principles become engineering foundations, and hopefully that's where we're headed with this. Thanks.
All right. Okay. Did you? Yeah. Yeah.
You can do the slider first.
You ask because you're standing up.
Okay.
Yeah.
Great talk. Have you thought about applying these models to societies like polities or states?
Yeah. So the work I didn't mention is there's a whole world of trying to think about urban scaling. People like Louise Bentoncourt and Jeffrey West and Vicky Young and Andrew Steer, a whole bunch of interesting people doing really exciting work there. And there it's interesting because maybe urban environments look more like ecologies than they do organisms, right? You and I can each decide to start a new sort of restaurant in Kyoto. Kyoto doesn't have much to say about that other than some basic permitting. You and I could have a good or bad idea, competing restaurants, all of that. And that's less like companies, which are often allocating resources internally to make a collective decision. So to go up to the state level, there I think we do start to recover organism ideas again, because it has this more unified perspective. There's interesting work on country scaling. So you can look at things like GDP against population size. There's a nice scaling relationship there. Those are places where there's a need for more mechanistic thinking. I mean, even in the company work, not all of those exponents are pinned down yet by fundamental theory. We're using them as empirical observations to build other theories. The same is sort of true for the polity or country level. People have done some really exciting historical work there as well that would be of interest. Yeah, thank you.
All right, let's ask the top question from Slido. It says, amazing talk, I agree. Does constraints on energy efficient information processing mean that digital ALIFE and hard robot ALIFE is impossible? Or can ALIFE find a way?
I think ALIFE can find a way. And I think there are lots of cases where we're seeing the thing I didn't say is that our computers are more inefficient, but if you plot the trajectory over time, they're headed in the right direction of efficiency. I expect them at some point to start to overlap with the molecular world. It just may take a bit more time. So I think ALIFE eventually will, I'm talking to the screen as if the screen can see me. ALIFE eventually will, I think, start to, will eventually start to have the same sorts of efficiencies as organic life. Thanks, great question.
All right, maybe the last one we'll go with the one that just got suddenly a bunch of votes. So does the kinetic versus diffusivity relation rely on assuming well-mixed innards? Could an organism deviate from the curve if its insides were spatially structured?
Yeah, so I should have gone into that in more detail. What we're actually doing there is solving the spatial gradient of both the products and substrates. And there you find internally there's these strong gradients in both. And that's the thing we're optimizing over. So it's actually, we're assuming that the inside of a cell is not well mixed. And that's what gives the scaling relationship. In that same paper, we go to eukaryotes, which then start to have intracellular transport processes, which are basically doing different types of mixing processes. That's essential for them to get bigger than the largest bacteria. The largest bacteria run into thermodynamic constraints associated with this. I didn't talk about any of that, but in that archive post, we do the same sort of optimization for how much mixing you want to add in eukaryotes, and that's essential to predicting the single-cell eukaryote scaling relationship. Great question.
Okay, I think we have time for just one more question.
Okay, sort of two parts. About the ribosome. So that presupposes, if we're looking elsewhere in the universe, that there are things like ribosomes, DNA, but that, for example, in artificial life, von Neumann imagined other mechanisms of self-reproduction by like scanning and copying, things like this. So that kind of analysis wouldn't apply. So that's one thing I'd hope you'd respond to. And the other is, as far as like life detection, you mentioned Sarah Walker and I guess Lee Cronin, molecular assembly index, if you could comment about that.
Yeah, perfect. So on the first point, yeah, I mean, I think there's many peculiarities about our translational system. I mean, for one, we do it in this multi-step process. That multi-stepness is something I don't think you always need to do. There may be good or bad reasons for that. There's trade-offs. I don't think we understand that full trade-off space. People have written endlessly about some of the proposed trade-offs and so forth. And then, yeah, I think you could have other sorts of structures for information storage and so forth. In that one paper, we argue about why linear information, and there's some good reasons for that, but it's important to write down theories for the full space of different types of information storage and see really where you have trade-offs and so forth. So I agree, I think that's a really important area. In that cell encapsulation paper, we really do just generalize to catalysis and autocatalysis. And there we're just talking about the volume of things and their speed. That's a macroscopic parameter that could come from a lot of microprocesses that we don't specify. And even there we get constraints, and that's really the hope, is we want models that don't depend so much on the particularity. So some of that's in there. So I'm involved with assembly theory as a concept and so forth, and I'm on some of those papers. And I think it's another example of, I don't have my picture, but it's another example of that top level sort of principle, right? Which is to say, just asking how assembled something is and asking how it was assembled, how it reuses subpieces and so forth is an exceptionally general idea that doesn't require what the pieces are, doesn't require knowledge about what the pieces are. It's developed to think about that. Its whole goal is to try and not think about what the subpieces are, but just ask how assembled an object is, independent of the alphabet you're using to assemble it. So I think that lives in that same top level of optimization principles, or at least it's a metric or measure for that top level. Yeah, great questions, thanks.
Okay, that's it for questions, I think. So let's thank the speaker again.